Greetings, grade 12 learners of life sciences. Welcome back to our life sciences class. We are carrying on with the revision for paper 2. Our topic will focus on genetics and inheritance, which you already know that it is one of the topics that weigh the most marks in paper 2, having the marks total of 48. So it's one of the topics that you need to make sure that you can get some marks there. So the first thing is that in genetics, you must know your terminology. Your Mind the Gap study guide, it gives you the definitions clearly of the different concepts in genetics. And once you can understand all the terms, then it's easier to go to the application questions. Now we're going to start our lesson by looking at a question that is applying the different types of dominance. Now you will remember I mentioned earlier on that we have got 3 types of dominance. We have complete dominance, whereby you will have a dominant allele that is masking the effect of the recessive allele. The recessive allele is only expressed when it is present in an organism in a homozygous condition. Then you get your incomplete dominance. Your incomplete dominance is the one where you have got 2 different phenotypes and they come together during fertilization. The alleles of those characteristics, they come together and they will give you an intermediate phenotype. And then you have codominance. Codominance is when the 2 alleles, they are both present in the phenotype of an organism. So if you think about human beings, in our blood groups, the blood group AB, it's because the 2 alleles, allele A and allele B, they are existing together, and that's why it gives us the phenotype type AB. So you need to know the difference there. Let us move without wasting time. Now remember, I told you that you must expect to read in a life sciences paper. This is a typical exam question paper, where here it says that a gardener had orange flowering plants in their that were growing in their garden, and she collected seeds from the plants and planted them. And when the plants of the F1 generation matured, she found that some of the plants produced red flowers and some produced yellow flowers. What did we start with? We started with orange. They're saying they had orange flowering plants, but when they produced the F1 generation, some of the flowers, they had red flowers, while some had the yellow flowers, and some produced the orange flowers. Now it says when red and yellow are mixed, orange is produced. So what does that mean? It means that orange is the intermediate. It's the intermediate phenotype between the red and the yellow that are crossed together. So let's proceed with the rest of the question. Now there it shows you a graph that were of the plants that were produced for each flower color. There were 942 of the plants were having red flowers, and 1,920 had orange flowers, and the ones that had yellow flowers, it was 1,018. Now, the question that was asked from this first question was asking, name the type of dominance. So they had to name the type of dominance for this. And the type of dominance there is incomplete dominance, Why is it incomplete dominance? Because orange is an intermediate phenotype. So neither the alleles yellow or red is dominant to the other, so they form an intermediate phenotype when they are existing together. Now, what is the ratio of the red flowers? So we come to the numbers. Red flower was 942. So it's red flowers to orange flowers. So we go look, what was the number for the orange flowers? It was 1,920. So it's 942 to 1,920. If you've got your calculator, you cannot leave this ratio as it is. You have to simplify it. So if you say 942 divided by 942 is to 1,920 divided by 942, that should give you a ratio that says it's more or less 1:2. So that would be the simplified ratio. If you left it at 942 is to 1,920, you wouldn't get the marks. Let us move to the next slide. Okay. Now, in this next slide, it's still applicable to the same question, but now it says you must calculate the percentage of red flowers that were produced in the F1 generation. So where is the red? We have got 942. But before we can work out the percentage, we need to know what was the total number of the plants that was produced. So if you look here, we have got 942 plus 1,920 plus 1,018. It gives us the total of 3,380 plants. Then to work out the percentage, remember here, look at the marker location. It says it is out of 3 marks, and therefore you need to be able to know how do you get those 3 marks. So you've worked out the total. So what we are going to give you the marks for is to know that we take the 942, which is for the red flowers, we divide it by the total. So you get a tick for that. So then the other tick, because we are saying calculate the percentage. So when you calculate the percentage, you always have to times by 100. Then you leave your answer. We usually say that your answer must be left to 2 decimal places. So it should be 27,87. That is where we usually ask for but the question will stipulate whether you must leave it into 1 decimal place or you can leave it to 2 decimal places. But usually we say that our calculations must be left to 2 decimal places. Now here again, this is a question that is asking you now to do a genetic cross. Now you will remember I said here you've got show marks, 2 marks that you can get easily here. Firstly, you have to indicate the P1 or the first parental generation, indicate the phenotype, and then you indicate the genotype. So it says in this question, use a genetic cross to show the crossing between 2 orange flowering plants. So you are crossing orange with orange, and therefore it means that because we said this is incomplete dominance, we are going to use the R for the red flowers and the yellow for the yellow flowers. That means that when we have orange, our orange must be RY, right? So orange is represented by RY, that is the genotype, and then we have meiosis, which is going to form the gametes, which will be the R and the Y from that parent, and then you have another R and Y from that flower. Okay, then you have fertilization. Now the R and the R there will give you capital R, then you do this one, and the one in blue is going to be RY, and then the Y and the R, you can put them as RY again, just for the sequence of the alphabetical letters, and then the Y and the Y. So your genotype for F1 will be RR for the green, and then for the blue, it's RY, and then for the black here, it's going to be RY again, and then for the last one, which is indicated across in red, is YY. Now, I said your two show marks, if you give us P1 and F1, we're going to give you a mark there. If you give us meiosis and fertilization, that is another mark. But they must go in the correct sequence there so that you can get, you can't have fertilization before meiosis, because it must be meiosis that must take place, and then the gametes are formed, and then fertilization. So those 2 marks, all the learners should get those 2 marks there. All right. And then your phenotype, when you interpret now your genotype, if it's RR, then it means that you have red. And then these 2, they are RY. RY means that you have got 2 orange, and then the YY means that you've got 1 yellow. So that would be your genetic cross. So please make sure you get those 2 marks there. Genetics, dihybrid cross is a little bit complicated, but what we have found is that the learners are not able to compute the gametes from a dihybrid cross. So for example, if you got a tall green plant, dihybrid, it means that we are now looking at the inheritance of 2 characteristics at a time. Di means 2. So it's an inheritance of 2 characteristics. So if we are looking at this example, we've got a tall green plant. So we are looking at 2 characteristics. In this case, it is a height of the plant plus the color of the plant, or the flowers rather. So it's a color of the flowers. Those are the 2 characteristics that we are getting there. Sorry of my writing. So if we're looking at a tall green plant, you will see in this case, maybe it said in the question that it was heterozygous for both characteristics, meaning that you have got a capital T and a small t for the height, and then you get a capital G and a small g. So it means that it is heterozygous for tall and for the height and for the color of the flowers. Now, how do we do the gametes now? Now, if we look at the height from here, there are 2 alleles there. There is a capital T, which we have indicated there, and there is a small t, which we have indicated there. Then on the second characteristic, we have a capital G and a small g. This is now computing the alleles. So when we use a Punnett square to do the computation there, we can have, because our characteristic that we started with was the height and then the color. So when we compute the gametes, we are always going to start with the letters representing the height and then the letters representing the color of the flowers. So if we cross the T and the G, we are going to get T, capital G. Then we cross this small t with that capital G. Then we have lowercase t and capital G. Then we cross this capital T with that small g over there. It's capital D, it's capital T, small g, and the same way. So these would be the gametes from that one parent. So you get 4 different gametes, or in this case, yes, it is 3 different gametes because these 2 are the same, but you can write all 4 of them for you to get all the marks in the test or in the exam. So this is how we cross them if you do the crossing without doing the Punnett square, and then you would get capital T, capital G, and then you get capital T and the small g, then you get small t and the capital G, and then you get a small t going with the small g, like we have done in the Punnett square. So that is very important. Other than that, that is the usual thing to be asked to do the gametes, or know how many different gametes are there in a dihybrid cross. Now let us look at this example quickly. It says Hunter's syndrome is a sex-linked disorder. Now that should tell you something. Once it says it's a sex-linked disorder, it means that the inheritance of that disorder, it occurs on the allele that is found on the X chromosome. So we usually say it's sex-linked or X-linked because the allele is carried on the X chromosome. Now in this example, they are telling us that it is a recessive allele. It's not always a recessive allele. It can be a dominant allele carried on the X chromosome. But for this example, it tells us that it is a recessive allele, and that recessive allele, because it's recessive, we represent it with a small letter T. Now, the diagram below shows the inheritance of Hunter's syndrome in a family. First of all, what type of diagram is represented there? You should know that this is a pedigree diagram, all right? And what you should know is that once the squares or the rectangles, they represent the males, and the circles represent the females. And once the squares are shaded, it means that they will have a particular condition. In this case, the condition is Hunter's syndrome. Once you have got a shading, it means that the person is having a particular genetic disorder. And then once it's not shaded, it means that that person is not having that disorder. I want you to be very careful with the pedigree diagram. Use the phenotype that is indicated here. Don't use your own one. Try to avoid writing such things as normal. Here in this example, they said unaffected male. So if they ask any of those squares that are unshaded, you would say it's an unaffected male. But you can't say it's a normal male. It's not given in the key like that. So you must use what is in the key so that you are getting all your marks there. Let's quickly go through to the questions. Give the phenotype of Peter. Where is Peter? Peter is here. What do we see about Peter? Peter is a male because it's a square. And Peter is shaded, meaning that Peter is having Hunter's syndrome. Okay, so Peter is a male with Hunter's syndrome. Your syndrome. So it's important when you are doing pedigree to indicate the gender. That is very important. Possible genotypes of happy. Where is our happy? Our happy is right here. What do we see about happy? Okay, happy is having, it's an unaffected female. But what is important is also for us to check that happy is married to Ben, or they are in a relationship, and they have got two children, which is Jack and Patricia. we can see that Jack and Patricia, they are both is not affected. So they have to be X capital T Y. We are still wanting to find what is the genotype of Happy. Now the children of Ben and Happy, they do not have Hunter's syndrome. So it means that if Ben does not have the allele for Hunter's syndrome to give to a daughter, then it means that Happy can only be X capital T, X, maybe even small t, or they can be X capital T or X capital T, like that. So it means that in this, sorry, that one is not a possibility because, sorry, can I rub that one out? That one is not a possibility because none of the children have got the syndrome. So it means that Happy has to be X capital T, X capital T. So that whenever they pass an allele, it must not affect the son who is Jack there. That means that Jack will be getting the X capital T from the mother and the Y from the father. So that is the possible genotypes of Happy. Let me be quick. Oopsie. Okay.
Don't worry, I'll cut it out.
Okay. Ooh, why it went to that side? Okay. Then for 2.74, it says why Lucky's condition cannot be used as evidence that John is his father. So if you look at John, John is a male which has Hunter syndrome. So John has got the recessive allele. So they've got X small t, Y. And then Lucky is also a man with Hunter syndrome. They also have X small t and a capital Y. So therefore, because both of them carry the recessive allele, then it means that they both have the Hunter syndrome, and therefore Lucky has the XTY as I've indicated, and he inherited that XT from her mother, because you get X chromosome from your mother and the Y chromosome, because he is a male. So he couldn't have got the X capital T from the father, because the father is also affected. That's why we cannot use that evidence as that John is his father. I will end it there for today. Thank you very much for listening. We have the comment section where you can write your questions or you want things clarified. I may have made mistakes, but we can correct them in the question section. Thank you very much for listening.